Hi, I'm Jay Hurst. Keep an eye out for a new episode of The Devil's Lettuce every Monday, where I'll interview industry professionals and like-minded people who are leading the way in the cannabis industry. Justin Sinclair is a naturopath, consultant and university lecturer who specialises in phytochemistry and is a passionate medical cannabis advocate. Justin is a research fellow at the National Institute of Complementary Medicines and is head of the Australian Medical Cannabis Research and Education Collaboration. He also sits on the Scientific Advisory Board for United in Compassion, which is an Australian charity formed to continue advocating patient access to full-spectrum herbal medicinal cannabis. Justin, could you elaborate on that intro and let the listeners know how you got into your field?
Yeah, certainly, Jay. It's a bit of a long story, but basically I had the opportunity when I was a little bit younger to go over and live in the United States. And as everyone I'm sure is aware, the United States has been investigating and utilizing medicinal cannabis, at least within various states, such as Colorado, California, Oregon, Alaska, and others. I think there's currently 13 for medicinal purposes, and that definitely started to get my interest. But when I first started getting my research interest was probably when I was in my master's degree. I did my master's with the Sydney University Faculty of Pharmacy, and was investigating morphological structures of different plants, particularly trichomes. And of course, cannabis was not something that we studied back then, but it was a plant that was known for having an extensive phytochemistry within its trichomes, and it just started to pique my interest because as a pharmacognosist, which is my main background, we study drugs that come from plants, and of course a large amount of the medications that are currently used in modern medicine are plant-derived. And that just piqued my interest, and I started going down the rabbit hole from all sorts of different scientific disciplines, from botany and morphology, all the way down to ethnopharmacology, which is a passion of mine, which is the study of how different cultures have used plants or other substances for medicine throughout our ancient history. And also horticulture, the cultivation aspect of it. It's quite a unique plant in contrast to many others, being that it's affected by things like photoperiodism. And I just, everywhere I turned, I started to learn something new. And the thing that baffles me, I guess, is that when you consider, that there's about 240,000 species, I think, of flowering plants on the planet. And we've really only scientifically studied less than about a tenth of those for their medicinal virtue. And here I was learning so much about just one plant, one, genus, Cannabis sativa, and subspecies sativa, Cannabis indica, and the variability that occurs just within this one genus. And I was completely overwhelmed, and I have been studying it ever since. So solid research I've been doing for about the last 10 years, but an interest in the plant for at least the last 2 decades.
So we had the reefer madness propaganda back in the 1930s, and a lot of Australians are obviously still under the impression that cannabis is still the devil's lettuce. So could you start by explaining what the endocannabinoid system is?
Yeah, certainly. In short, one thing that's really important to remember is that we don't have an endocannabinoid system because of cannabis. We have an endocannabinoid system because we produce certain compounds that interact with the system. And it just so happens that many, many plants, not just cannabis, produce compounds that can also interact with this system. But the fascinating thing, I think, for me is that even though, as you rightly pointed out, in the reefer madness era of the 1920s, 1930s, particularly in the United States, which then, formed the basis for all of the prohibition that we currently see worldwide. Science was still investigating, so much about the plant. We didn't even know the main chemicals within the plant, which were, delta-9 tetrahydrocannabinol, CBD and all of these others that roll off people's tongues these days. And they weren't actually even identified until 1964 by Gayoni and Meshulam over in Israel. So the interesting thing about it is that the research into this plant actually discovered an entire neuromodulatory system that we're still only coming to grips with today. It has very, very far-reaching implications, particularly when it comes to our understanding of various diseases. But yeah, it's not just getting back to, I guess, on topic, the endocannabinoid system. It's been something that's been evolving for a long period of time. We've been able to find evidence that it's been within different animal types for at least 500 million years. So this is something that's not just shared by that humans have. It's something that all sorts of other animals do too. So other vertebrates and fish, amphibians, mollusks, leeches, sea urchins, and of course mammals. Insects don't have it. But it's just this, it has incredibly far-reaching implications for disease when it comes to, I'll give you an example, is autoimmunity. Autoimmune disease is an excellent example here because current medical research has a very limited understanding of what causes the condition. And because of that, because we don't understand cause, we have a very, very reduced application when it comes to management and treatment of these conditions. And so they're largely treated symptomatically. And what, I believe the endocannabinoid system is, and our understanding of it as it starts to unfold and evolve, is that we will have a much better understanding of how some of these conditions and diseases start to be perceived. And that's at least one of my hopes. I guess a concern is that the endocannabinoid system is still not being treated, I think, with the respect that it probably deserves, particularly by some in the medical profession, because it's actually not included in most of the medical curriculums within Australian medical schools, which is concerning. But to get back on track, we'll break down the endocannabinoid system and have a look at it, maybe to give some scope and just a little bit of background to it. Like I said, we didn't actually discover Delta-9 THC from the plant until 1964. And then research started to be generated, but most of it came from Israel because they were quite open-minded and didn't, jump on the reefer madness bandwagon. But Meshulam and Gayoni and I think Braun, one of their colleagues, they chemically synthesized it in 1967, and that allowed for a lot more research to actually occur because now that they could synthesize it, it was much more reproducible. I think it was in 1988, they identified that THC was actually stereospecific. And what that actually means is that it needs a certain receptor to bind to. And so this is when they started thinking, well, this is really interesting because what they were trying to do at this time, they identified these constituents, but they were trying to work out the mechanism of action by which it worked on the human body. And then later that same year, one of their colleagues, Devane, and I think Matsuda, they actually identified the first cannabinoid receptor, and this is known as the CB1 receptor. I'll talk about that a bit more in a minute. And so that's the second piece, the main piece of that puzzle was, okay, well, here's a compound, how does it work? And now we've identified a receptor that it actually binds to. And so this idea is, kind of think of like a lock and a key. Within the body, there's tons of different receptors that are locks, and there are all sorts of different compounds that we either produce inside our body or that we maybe ingest from food or drugs, other medicines that work as keys. And those keys bind to the lock, and they can open the lock and cause a physiological outcome. And so this first cannabinoid receptor identification in 1988 started, a huge influx of research interest. And then in 1992, just 4 years later, the first endocannabinoid, which was again identified by Devane, they found this endocannabinoid. Just means endo means within, and cannabinoid obviously a compound that is mimicking the cannabinoids that are found in cannabis, except that we produce it endogenously. We produce it inside the body. And this was called anandamide. And interestingly, it was named anandamide after the Sanskrit word for bliss or supreme joy because of its cannabimimetic effect. It mimics the psychoactivity of cannabis and THC. And so the next year, again, research was exploding. They found the second cannabinoid receptor. This was the CB2 receptor. And then in 1995, the second endocannabinoid, which is known as 2-arachidonyl glycerol, or 2AG for short, was discovered. And so why I wanted to highlight that is just to show that cannabis research has not just been important for studying clinical indications and using it for pain and all these sorts of other conditions, but it has actually forwarded anatomy and our anatomical and physiological understanding of the human body. And you would think, in the 90s and 80s that modern medicine has mapped and found everything, but it's certainly not true. And we're still learning more and more about the endocannabinoid system every day. But then if we focus on what are the components of the endocannabinoid system, well, we've talked about two of them. So we've talked about the cannabinoid receptors, which work as the lock. We've talked about the endogenous ligands, which is just a fancy name for the endogenous compounds that work as keys that fit inside those locks and trigger a physiological response. And so the cannabinoid receptors, the CB1 and CB2 receptors, are the types of locks, and of course there are more implicated now. These are not to get too technical, but these are a G protein coupled receptor that the cannabinoid receptors are, and there are of course other G protein coupled receptors within the body. And currently scientists are actually proposing that certain other G protein coupled receptors might actually belong to the cannabinoid receptor family, and so they're doing a lot more extensive research into that.
Just to interrupt you there, for people who don't know, and also myself, what's a G protein receptor?
Okay, so G protein coupled receptors they're basically in a nutshell, kind of hard to explain without visuals, but they're basically they have 7 transmembranes within them. It's kind of like, imagine that when that key goes into the lock and once it gets turned, there are also other components that need to be activated, and they go on to form a cascade of different types of outcomes. And that's something that's quite unique within the G protein coupled family. They work via inhibiting adenylate cyclase. And this is actually something that's important for inhibiting the conversion of adenosine triphosphate, which many of you might have heard of before, particularly maybe even in year 9, year 10 biology. And adenosine triphosphate is kind of like a cellular currency. It's something that is generated so that biochemistry can occur, so that different reactions can occur. And this inhibition of conversion of that adenosine triphosphate to its derivative of cyclic AMP, so your adenosine monophosphate, this then can have a flow-on effect with something called cyclic AMP and the protein-dependent kinases. And so where this kind of gets interesting is that this is some of these mitogen-activated protein kinases are what's basically involved around cellular responses. So when you start looking at the way a cell will respond to things like osmotic stress or inflammation, this therefore can assist in regulating a lot of the different cellular functions within the cell. So things like gene expression, mitosis. Mitosis is an important somatic or part of somatic cell division. So this is how cells actually replicate and replace themselves. Apoptosis is another one. So this is very interesting, particularly for cancer. And the reason that is apoptosis is just a fancy term for cell death. And what's interesting with cancer research is that some of these cancers can basically turn off programmed cell death, and that's why many of them are actually immortal cells, because they can just continue to replicate and replicate and replicate, and that's why the tumor can continue to grow and grow and grow. And the natural preventative mechanism to try and stop cancer is apoptosis. And so cancer cells can manipulate this so that they don't have to abide by the same rules and regulations as all the other cells. So this may be why cannabis can be useful for working, at least in some of the evidence I've seen in cell lines for cancer.
So is there a particular cancer that they work on, or is it just sort of still in the research progress at the moment?
Oh, yeah, still in the research, that's for sure. And what they're doing now is they're getting all sorts of different cancer cell lines, because that's something that's really important for the listener to know, is that, cancer is not a disease. Cancer is a cluster of many, many, many different types of diseases. And so the way that cancer is treated for breast cancer, the drugs that they will use for breast cancer will be very, very different to the drugs that they would use for prostate cancer, and different again to the drugs that they would use for breast cancer. And so they're doing lots of in vitro work, benchtop science to investigate the different compounds, obviously within cannabis, and how they might be able to work on cancer. But of course, there is a growing body of anecdotal evidence of patients that have been using cannabis and have been getting not only amelioration of symptoms, but some rather remarkable physiological results. But it's a completely new world when it comes to science, and there's lots of research interest in that area at the moment.
Could I ask, is there any Australian research going on to do with cell death and cannabis?
I must admit, I believe the Lambert Initiative may be doing some of that. They're based at Sydney University. Professor Ian McGregor is the academic director over there, and he has a great research team over there. I speak with Ian fairly regularly. Lovely man. But I'm not entirely sure of other research groups. I know that we're certainly interested in going into this space at Nickham at Western Sydney Uni. It's not just Australia. Obviously, there's Israel certainly is leading the charge when it comes to research worldwide. But it's growing. The research interest in cannabis is literally exploding at the moment. And I think that's obviously because of the changing public perception from that, going back to your original assertion, the reefer madness era. It's funny because I think it was Winston Churchill said, a lie travels halfway around the world before the truth has even had a chance to put its pants on, and nothing could be a better example of that than what happened, unfortunately, with poor old cannabis.
Yeah.
And so I guess that's basically the way that those cannabinoid receptors can work, and the other way that they do, so they work on those adenylate cyclases, they work on the mitogen-activated protein kinase. So those G-protein-coupled receptors, this is something that's quite closely associated with them. But the really interesting thing is that they modulate the activity of different ion channels. And so, things like potassium and calcium, particularly the voltage-gated calcium channels. And what this actually means in a broad example is that they modulate neurotransmitter release. Okay, so what's interesting is that some researchers have posited that there are more receptors for the endocannabinoid systems or the endocannabinoid system than there are for the neurotransmitters they modulate. And so when I originally opened up by saying that cannabis discovered this entire neuromodulatory system, this is exactly what I meant, because all of the different neurotransmitters from noradrenaline, acetylcholine, glutamate, serotonin, all of these very, very important receptors, but particularly noradrenaline, acetylcholine, GABA, and glutamate. What can happen then is that through the process of this modulation, they can inhibit either excitatory neurotransmitters or inhibitory neurotransmitters. And this has a significant impact on the outcome of physiology within that person. And so it's incredibly exciting when we start to understand that, and particularly from my background, because when we talk about health, we talk about homeostasis. And homeostasis is this concept that the body is in perfect balance, that, everything is working as it should be, and therefore health is being able to be achieved. And so when we start to look at health within the human dynamic, it's this constant ebb and flow between, maybe deficiency of certain things or excess of certain things, and then your body takes corrective action to try and bring it back to this homeostatic balance, this resting tone of the endocannabinoid system. And it really does seem to me that the endocannabinoid system is the requisite example within the body of how homeostasis is maintained. And that, I think, has far-reaching implications for health and disease management well into the future. But that's basically the main ways that those receptors work. And it's the endocannabinoids that we then produce and that work as keys in those receptors. So they've recently done a study which I thought was quite interesting, showing how I don't know how many of your audience may be exercise, that exercise a lot, maybe the joggers, and you've probably heard of runner's high. So you start, exerting yourself physiologically and exercising, and you start kind of getting that numbing action. You start feeling really good. And scientists had always believed that this was just the sole domain of endorphins, which are basically natural opiates that the body produces. But what we've actually found now is that we also produce anandamide when we're going through that exercise process. So it's really making you feel quite good, not just the endorphins, but the anandamide as well. And so our body constantly is producing these endocannabinoids on an as-needed basis. They're not necessarily, at least within the current scientific literature, these endocannabinoids are not stored within synaptic vessels, or vesicles, much like a lot of those other neurotransmitters like noradrenaline, acetylcholine, that I previously mentioned. They're generally stored within vesicles. But currently what's super interesting actually about this is that these endocannabinoids are basically manufactured from the postsynaptic terminal, from the membrane lipids and fats that are contained within there, and they're manufactured on demand. And again, being difficult to describe this without a diagram, but the reality is that most axon activity neurologically is one way. It travels from the presynaptic terminal down to the synaptic end bulb, the action potential, and that triggers the release of all of these neurotransmitters. And these neurotransmitters cross a very short little area of space called the synaptic cleft, and then they bind to receptors on the postsynaptic terminal, and then that continues the action potential, the nerve impulse. Now, what's interesting here is that this is kind of that one-way process, but what endocannabinoids do is they work by a process called retrograde signaling. So they're actually manufactured on demand, particularly 2-arachidonyl glycerol, that 2-AG, and it goes back across to the presynaptic terminal and turns the key to the cannabinoid receptor there, and that inhibits that neurotransmitter release. And so this is giving us a huge understanding into how this neuromodulatory system works. But that's actually only two parts of the equation, because actually we've got another part of that equation, which is the enzymes. And there are enzymes that are needed to manufacture and to break down our endocannabinoids. And so not surprisingly, anything that could impact on the amount of enzymes that are produced or that are what we call synthetic. So these are the ones that are required for synthesizing anandamide or synthesizing 2AG, actually making it available. Or then there are those compounds like fatty acid amide hydrolase. And the listeners might be interested to know, when they hear ASE or A's on the end of anything, generally that can be meaning that it's an enzyme. And this fatty acid amide hydrolase, for example, breaks down anandamide into different component parts, largely arachidonic acid and ethanolamine. And so if you have a deficiency in either of those types of enzymes, ones that either produce endocannabinoids or break them down, then you're also going to see a disturbance within the endocannabinoid system also, because that's going to have an effect on how much anandamide or how much 2AG is going to be available. And interestingly, they started doing studies where they started to reduce the breakdown of things like fat or the amount of fatty acid amide hydrolase. And obviously that means that more anandamide is going to be floating around and available. And this is quite a, big interest currently within pharmaceutical industries and others. And so this dynamic interplay between the endocannabinoids, the GCPR, the G protein coupled receptors, the cannabinoid receptors, CB1 and CB2, and the enzymes that actually produce the endocannabinoids are what basically make up our endocannabinoid system. And the cannabinoid receptors themselves can also be broken down quite a bit by looking at the CB1 and the CB2 receptor. The CB1 receptor is actually found largely within the central nervous system. And this, it's everywhere through the cerebral cortex, the basal ganglia, the substantia gelatinosa of the spinal cord, cerebellum, the hippocampus, all sorts of different areas. And this is the one that is largely associated with the psychoactivity. So when you utilize cannabis medicinally or otherwise, this is going to be where it binds to. And this is what causes all of that psychoactivity, is through the CB1 receptor. So that CB1 receptor activation is what's going to then inhibit the neurotransmitter release of all of those, glutamate, GABA, dopamine, acetylcholine, noradrenaline, of both those excitatory and inhibitory synapses. And this is where a lot of the potential relief of pain and things like that from cannabis compounds comes from. Whereas the CB2 receptor is actually a little bit different, because this is mainly found within the, looking at places like the tissues of the immune cells. Sorry, I was having a bit of a brain fart there. But the immune cells, so places like the spleen, the thymus, tonsils, right throughout the gastrointestinal tract, they're all through there, but also found within the cells of our immune system. So the macrophages, kind of like the little baby Pac-Men that go around and destroy any invading pathogens, and monocytes are much the same. Natural killer cells, which are kind of like our special forces that are running around not only killing pathogens but cancer cells, and neutrophils, which are our kind of like our first line responders to bacterial invasion. So the CB2 receptors are rife throughout the immune cells. But the interesting thing, of course, is that because the CB2 receptor is not found a lot within the central nervous system, it's not noted as having any major demonstrable psychoactive activity, but certainly has a very important role in local analgesic effects. And one of the interesting things there is that neuroprotective effects and anti-inflammatory activities have also been correlated with CB2 expression within the tissue. And CB2 expression itself is actually enhanced. And expression is just a fancy name for how many locks become available physiologically. So it gets enhanced. The number of locks that are required are enhanced because of inflammation. And inflammation is the body's only way of dealing with all sorts of different things. So when you get a cold or a flu and you sit there cursing the person that gave it to you or the flu itself, understand that most of the symptoms that you're getting are actually our body's response to try and get rid of it. So inflammation is it's an important mechanism for repair within the body, but of course, particularly in chronic illness, chronic inflammatory conditions, this is just an ongoing problem, and obviously a major cause for concern for the patient and the medicos treating them, because as we've said previously, for many of these conditions, we don't understand the cause. So, the treatment is largely symptomatic. But I think that probably in a nutshell is what the endocannabinoid system is, and that's where the different compounds found in cannabis can interact with those various receptors and have that cannabimetic activity. So mimic the activity of our own endogenous endocannabinoids.
Okay, and could you let us know how listeners can follow your work and stay in touch with you?
Yeah, look, I must admit I'm probably not the best on social media, but I do have a webpage and my website. My website is www.tmconsultancy, all one word, so that's Tango Mike Consultancy, all one word, .com.au. And that's kind of just where I load up any podcasts that I've been doing, maybe like this one, and other educational resources. And that webpage has got emails, and generally I do have a Facebook page, just a personal one, Justin Sinclair, that I'll occasionally get on and post some things of interest. But generally I'll use my website to promulgate any interesting information.
Okay, excellent. Thanks for coming on today, Justin.
My absolute pleasure. Thank you for the invitation, and also for providing a platform for education, because I must admit, patients and carers particularly are clamoring for more information, and I'm really supportive of the work that you're doing.
Thanks for listening to the Devil's Lettuce podcast. If you enjoyed this show, we'd love you to subscribe, rate, and give us a review on iTunes or wherever you listen to your podcasts. If you have a suggestion for a guest on the Devil's Lettuce, head over to thedevilslettuce.com.au and click on Suggest a Guest. Please do not take any information from the Devil's Lettuce or its guests as medical advice. Thanks for listening. See you next time.